Okay, all right. I'm going to kind of continue on the same talk of what the molecular foundation of life has to say about Darwin's theory of evolution. And it's interesting, if you go back through the history of science, you see that science often advances very rapidly because new technology has been invented. For example, when Galileo looked into a telescope, which was pretty new for his time, we learned a lot about the universe. And when the microscope, working microscope was invented, then biologists discovered a tiny world that nobody knew was there. Little insects with compound eyes and all sorts of crazy stuff. Well, it turns out that we are living through a revolutionary phase in biology, similar to when the microscope was invented. And the reason, again, is due to new technology. In particular, the ability to sequence DNA. We've heard that DNA is kind of the repository of information in our cells. And it's the exact sequencing of letters, A, T, C, and G, over 1 billion or more positions that helps determine who we are and our physical traits. But until really pretty recently, past 10, 20 years, the technology wasn't available to be able to sequence enough of that easily enough so that you could do it in multiple instances. The technology wasn't available to be able to determine the sequences of those letters. One could do spot sequencing here and there, but not the amount that would be required to determine a mutation that had been helpful, see how it spreads through the population, and what effect it has. Now, however, we can. So here's DNA, as you all recognize, and this represents some of the technology that separates one kind of base from the other, and this puts them in order, and then you eventually read out the exact sequence, T, C, T, T, G, G, and so on. And that's the topic of my book that was published just this past February, Darwin Devolves. And there are 3 key concepts that I discuss in the book, something called the first rule of adaptive evolution, the principle of comparative difficulty, and the family line. But because of our restricted time available here, we're going to have to deal with just the most important one, which is the first rule of adaptive evolution. And the other 2, well, you'll just have to buy the book. Hey, I got kids, Okay, so the first rule of adaptive evolution, what the heck does that mean? Well, the history behind that rule has a lot to do with this fellow here. This is a man named Richard Lenski, and he's a professor of biology at Michigan State University. And about 30 years or so ago, he had an idea for an odd kind of experiment. What he would do is take a bacterium called E. coli and put it in a flask. And for replicability's sake, he had 12 flasks and he put a portion in each. And he had enough food in the flask in the form of dissolved sugar for the bacteria to go through 6 or 7 generations. And since bacteria reproduce so quickly, that just kind of took overnight. So he just let the flasks be, kind of shook them so they'd be stirred up. And next morning he came in and he took an aliquot, a little drop from each flask, and put it into another flask with also the same amount of food so that it could go through another 6 or 7 generations. Then he came in the next day and did the same thing, and the next day, and the next day. And for the past 30 years, he has conducted this for what is now about 70,000 generations of E. coli. And they're so small that, in fact, over that time, tens of trillions of cells had been produced. And he did this just to see what would happen, how they would change, how they would adapt, how they would evolve. And in the beginning, in the year 1990 or so, he saw that in one flask, some bacteria started to grow faster. And quickly they kind of overtook the culture. And then a little bit later, they started to grow faster again and overcame the culture. And so he was observing in real time Darwinian evolution, mutations coming along and helping the bacteria adapt and increase in its environment. But at that time, it was not possible to track down what these mutations were. The genome of E. coli is 4 million base pairs in length. And then it was just hopeless to try and track down changes in a few places out of that 4 million. So he continued for the time being and just kind of described what he saw, that every now and again a mutation came along, it would grow faster, and in the different lines, in the 12 different lines, it'd come along at different times as if random changes were happening in different places in the 12, and he would publish on that. It wasn't until about the year 2000 that they were able to track down one change at the molecular level. And this is the paper reporting that. And what they saw was that each of the 12 replicate cultures lost the ability to make a certain kind of sugar called ribose, or metabolized a certain kind of sugar called ribose. And so that gave them a clue to zero in on. It turns out it's known where different genes are in the genome of DNA. So they looked at just the region that contained the several genes that are needed to metabolize this sugar, ribose. And what they saw was that another piece of DNA had inserted itself in there and pretty much blown up the region, so that the ability to metabolize ribose was lost. And strangely, that helped the bacteria reproduce faster. So as you see, these changes may reflect beneficial mutations in these regions. Indeed, deletions of the ribose operon were found in all 12 of the evolved population. So this was, fascinating, because it shows that a beneficial mutation is not necessarily a constructive mutation. That sometimes getting rid of a gene or two can be helpful in some circumstances. And after that initial paper, they tracked down some other mutations in other genes, and those are shown here. And each of these bars, A, B, C, and D, represent different genes designated here and here. And the arrows point to places within the genes that had changed a letter from one to another. And the various arrows were from the various replicate cultures, those 12 replicate cultures. And you see that for each of these, there's a lot of different places where you can change the gene, and it would be helpful. All of these mutations helped the bacterium grow faster. And it turns out that this is the kind of result you'd expect if the mutations were helping by breaking or degrading the function of the gene. That is, the change makes the gene less effective, or less of it is produced, because it's easy to break a complicated machine, but it's real hard to improve it. Just like, if you hit your computer with a hammer, you could hit it in a number of different places and it would run less effectively or not at all. Very few places could you hit it with a hammer and it would improve it. So it's kind of like that here. And all of the genes he reported at that time got better, were beneficial, because they were losing function. Well, about 10 years or so ago, I was intrigued by Richard Lenski's work, and so I wrote a review paper for a journal called Quarterly Review of Biology, in which I looked at his work up to that time, plus the work of many other laboratories who were involved in experimental evolution of microorganisms, either bacteria like Lenski or viruses like a number of other laboratories. And what I showed was that all the results up to then, the great majority of beneficial mutations were, like in Lenski's case, those that degraded or broke genes. There were some that didn't, but most of them did. And so I always look for pithy ways to summarize results, because that gets more attention than otherwise. And so I summarize the results as the first rule of adaptive evolution. And the first rule of adaptive evolution is the following. It is to break or blunt any functional coded element whose loss would yield a net fitness gain. So I had to write in the obscure style that is popular in scientific journals. But what this actually means is that you should either break completely or at least degrade any gene or a control region for the gene whose loss would yield more offspring. For some reason, evolutionists call more offspring fitness. So a net fitness gain. And this I called the first rule of adaptive evolution, because it's a rule in the sense of a rule of thumb. This is what you'd expect to happen most often, most frequently, but not all the time. It's just a rule of thumb. And it's called the first rule because this is what you'd expect to happen first. The first thing that will come along would be a potentially helpful degradative mutation. And the reason why such mutations would come along first is easy to see. It's like hitting your computer with a hammer, and of course most of it will break, but suppose you hit it and it jarred something and it broke a circuit, and that somehow made the computer run faster, at least for some special applications. Well, the same thing with genes. You might, if a gene is going to be improved, there's only a few places in general that you would expect to be able to change to make it better. And that's because so many genes and the proteins they code for already work really very well. On the other hand, if you want to break it, you can hit it just about anywhere, because you can interfere with its function in many different ways. So if a gene could be helpful by being degraded or broken, then it will appear really faster, much faster than a gene that would help by having a new, improved, or additional function, simply because of statistics. It's a lot faster, a lot easier to break the gene. But some people say to themselves, how can you help something by degrading a gene? Why does it help to break something? Well, let's think about an example from our everyday world. Suppose that your life depended on your car getting a little bit better gas mileage, a little bit better. What could you do to quickly get better gas mileage? Well, you could decrease the weight of the car. So you could take off the doors or maybe the roof or windshield and so on and throw those away. And your car would get better gas mileage just because it's lighter. Of course, those things like doors and windshields are useful in many circumstances. But if your life depended on the car getting better gas mileage right now, then on the right, that would be the way to go. Otherwise, you wouldn't be around to enjoy the door or the windshield or whatever. Another way, this is a bit more obscure, another way to helpfully affect something by degradation is if you have a very complex, say, plant like a chemical plant where each of these pipes is feeding different chemicals into each other to produce products. And maybe just by shutting off a valve here or somewhere else or breaking the valve so it has to stay open or shut, then maybe it gives you more of the product that happens to be selling well at the time, and so you do better. Of course, now you can't switch back easily, but right now, right when selection is acting, you're doing better than everything else. Well, in the year 2016, the sequencing technology has come to flower. Sequencing has become much, much more easy than it was. It's kind of like the computer revolution. In the 1950s, computers were large and clunky and expensive, but now they're small and fast and cheap. Turns out it's the same with the sequencing of DNA. And sequences for all sorts of creatures have been done and put in public computer databases where anybody can access them. So not only has the sequence of human DNA been determined, but also dogs and cats and snakes and rice plants and wheat plants and bacteria and all sorts of stuff. Well, when in 2016, taking advantage of this technology, Richard Lenski and his colleagues sequenced the DNA of every 1,000 generations of each of the 12 lines of bacteria from his experiment. Now, it turns out he can actually keep some of the earlier bacteria because every 500 to 1,000 generations, he'd take some of the bacteria, put it in a test tube, and put it in a freezer. Turns out that doesn't kill the bacteria, but it kind of puts them in suspended animation. So he can later, maybe years later, take it out and determine what its DNA is. So in a paper in the Proceedings of the National Academy of Sciences, I think that is, he published the results of the 30 most highly selected mutations. In other words, the 30 mutations that helped the bacterium the most in adapting to its environment. And can you read that? Actually, it was in Nature. And what he saw Look how cool this is. Isn't this cool? In the first column are the different genes which had only point mutations. That is, only one letter had changed in those genes. But in the third, this is the number of different mutations that they saw across the 12 cultures. You see, the first one has 19 different places where you can mutate it and it becomes better. And even the least one has multiple changes where it's beneficial. So again, like the genes we saw a couple slides ago, this is the fingerprint of a process that is helping by degrading the genes that are being mutated, because you can change them at any of a variety of places. This was the first table that he published with about 15 genes. The second table in this paper was also of about 15 genes, and these are genes with the most mutations of other types. In other words, big regions of DNA might be inserted into a gene, or the gene deleted entirely, or things flipped around, and almost always that kills the gene. And you can see the number of different mutations that were isolated in the various genes, and all of them were isolated multiple times. So the long and the short of it is that after 50,000 generations, which is what it was when he published this paper, after trillions of bacteria have been selected, have lived and died, have spread and improved, all of the mutations likely were degradative. All of them involved decreasing or eliminating the activity of complex genes that were already there. None of them involved building new complex systems. So this kind of summarizes what our observations show now that we can detect mutation and evolution at the molecular level of life. We already knew since the 1950s that of those mutations that affect an organism, about 99% are detrimental. And evolutionary biologists would say, that doesn't matter because it's only the ones who survive, the beneficial ones, that will reproduce and continue. But now, because of the work of Lenski and others, we find out that of even beneficial mutations, helpful mutations, the great majority break genes or degrade function. And it turns out that, kind of like the second law of thermodynamics, this degradatory process is as relentless as the tide. It may be that a mutation comes along which is helpful, which doesn't break a gene, maybe even adds something. But when it does, the next mutation that comes along will quickly be a degradative one, and because they're just so much faster. And here's an example of that. During Lenski's experiments, he saw that one strain of the bacteria, one of the 12 strains, developed the ability to eat citrate in the presence of oxygen. Now, we don't have to worry about the details of that. Just remember that bacteria, E. coli bacteria, can already eat citrate, but only in the absence of oxygen. The only difference was that now they could eat it in the presence of oxygen. And they later tracked down the mutation that allowed that to happen, and they saw that here's the gene that allowed, that pulls citrate into the cell, and it turns out that an accidental change duplicated a portion of this, and it accidentally put a control element next to one of the duplicated portions that allows it to be expressed, turned on, in the presence of oxygen. So now that bacteria could eat the citrate, which turns out there was a lot of citrate for technical reasons. There was a lot of citrate in the broth, and that bacteria could eat it, and it rapidly took over the whole flask. The next day, Lenski and his students came in. They saw that one flask was really cloudy. Most of them didn't really get cloudy. The cloudy flask turns out to have a lot more bacteria in it than before. And this got a lot of press. Lenski has a red phone in his laboratory, and when he picks it up, it connects directly to the New York Times. So this got a fair amount of publicity. I myself would not call this a big deal because it just duplicated stuff that was already there. But it turns out it doesn't matter what this particular mutation did or did not do. Lenski's group and people who sympathize with him were touting this as a big deal. But if you look at subsequent research on this mutant, some of it was done by Lenski's students here, this fellow here, in their own lab when they graduated and became professors of their own. And they looked at what they called fine-tuning of that citrate mutation, the one that can now eat citrate. So they said, well, what changes in metabolism would help this new mutant do better in its environment? And they saw that one was the loss of a gene for something called isocitrate lyase. And so they looked for this mutation and saw that the gene was broken. And they looked for a couple of other mutants too in these two genes, and saw that they lost their function too, because it helped in a bacterium that was using citrate as a food to break these genes, because that would help them metabolize it better. So the point is that every now and again, a non-degradative mutation can come along, and that's fine, it's just probability, but the odds are good that, most of the changes will be degradative. So we can conclude that Darwin's mechanism is dominated by what I call poison pill mutations. That is, positively selected loss of function mutations. And that Darwin's mechanism works chiefly by squandering genetic information for short-term gain. It's important to realize that once this mutation occurs, because the mutants do better, they spread throughout the population, and the original bacteria are gone. So now you've got fixed in the population, all the population is missing the gene. And once the gene is missing, it's gone forever. It's not coming back. Well, I myself am a biochemist. And biochemists like cells and molecules. Hey, this is really interesting. But it's come to my attention that other people aren't all that interested in bacteria. So let's look at a couple of other creatures more well known in our everyday world. Starting with doggies, good old doggies. And as we know, dogs come in a whole bunch of sizes and shapes with curly hair and abbreviated snouts and short legs and so on. And it's been a while that Darwinists have pointed to dogs and said, look at this variability. This shows what selection and random changes can do. And not natural selection, of course, they were selected by breeders, but it's pretty analogous, because if nature wanted short legs or curly hair, it would probably select the same sorts of dogs that the breeders did. Well, it turns out that because sequencing is so easy these days, that the entire genomes of dozens of different breeds of dogs have been sequenced and compared with each other to see what gives them certain traits. And that's shown on this next slide. And it turns out that like the E. coli, getting various phenotypes or traits like short legs or curly hair or lots of muscle and so on is due to mutations in specific genes, most of which are loss of function or strongly indicated degradation of function. So even something that strikes us as a gain of something, there are some dogs that are highly muscled. They're really much tougher looking than other dogs, but it turns out that is due to the degradation of a gene that tells the body when to stop making muscle. Okay, oops, did I miss that? Here, oh yeah, sorry, skip that. Okay, another example of a creature we're more familiar with is polar bear. Now, polar bears are thought to have descended from brown bears, grizzly bears, over the past few 100,000 years or so. And again, just because of the advance of technology, scientists have been able to sequence the entire genomes of brown bears and polar bears and compare them for what genes were most helpful for helping the polar bear adapt to its environment. And the first paper reporting it showed that we assessed the impact of specific substitutions, and a large proportion of the mutations were predicted to be functionally damaging. And it turns out they write 50% here, but if you look at it a different way, it's closer to 75%. Because many genes have a couple of mutations, and 75% of the genes have at least one degradative or damaging mutation. So you say, well, polar bears are magnificent. How can that be? Well, it turns out if you want white fur, then you just break genes that are responsible for making the brown fur, the pigment in the polar bear. Polar bears eat a lot of seal fat. They survive mostly on blubber. They eat a lot more fat than do grizzly bears. And it turns out that breaking a gene in fat metabolism helps them to be able to tolerate that higher level of fat. So it turns out that you can get a lot of variety in a lineage that looks really cool, but when you look at it at the molecular level, where you have to evaluate Darwin's theory, those adaptations are occurring by breaking genes that were already there. So here's an image I want to you guys to see, and this is a photo that I found on the internet, and so it's true. And this is a couple of soldiers in a South American country, I think, and they had gotten information that there was a drug growing operation somewhere up in the woods. And so they were launching a raid to raid the drug facility. But somebody tipped the drug guys off to the impending raid, and the drug dealers blew up the only bridge on the only road that led to their compound. Now, from the point of view of the drug dealers, this is a beneficial change. Quite literally, it lets them live for another day, and maybe they can scramble out, take stuff with them, and survive and set up camp somewhere else. And this is literally what Darwinian evolution does in many cases. Let me give you an example. Everybody knows that malaria is a deadly disease. It still kills nearly 1 million people per year, mostly in Africa and India and so on. It's caused by a little single-celled parasite called Plasmodium falciparum, which is injected into people by the bite of an infected mosquito. And it turns out that the cell then goes and attaches itself to a red blood cell, enters the red blood cell, and it eats the hemoglobin inside the cell. And it uses that material and the energy from it to make about 20 copies of itself. And then that bursts open the red blood cell, and each of the 20 copies attaches to another red blood cell. And after the same thing, they burst out, and now there's 400 of them. And after another round, there's 8,000, and so on. And pretty soon they've eaten most of a person's blood, and that's one of the main causes of death. Well, it turns out that the malarial cell, when it attaches to a red blood cell, has to attach to a very specific protein. It can't just glom on anywhere. It has to have a specifically shaped docking area. And that's where it hooks onto and uses that to enter into the cell. Well, it turns out there's a group of people in West Africa that are immune to malaria. And when their genome was sequenced, it turns out that they had deleted the gene that coded for that docking protein. They literally blew up the bridge so that they would not be invaded by the malarial parasite. And from an ordinary point of view, we would say, wow, look, these people, they are resistant to malaria. Isn't evolution amazing? You know, an Englishman goes in there and gets bitten by a mosquito and, goes toes up in short order. But these folks, they are resistant. But it turns out that when you look at the molecular level, again, you gotta look at the molecular level. It turns out that it's an improvement, all right, in the circumstances, but it's one that's caused by degradation. Okay, now, so that was the first rule of adaptive evolution. And as I said, we're going to skip over the other ones, but you're probably sitting there saying, yeah, I see your lips flapping. But what do real scientists say about this? Everybody knows that if you don't think that Darwin's theory is correct, you can't be a real scientist. Well, we have an opportunity to see that because Science magazine, one of the 2 top journals in the world, reviewed my new book, Darwin Devolves. Interestingly, they reviewed it, they assigned it to 3 authors, to 3 reviewers. I've never seen 3 reviewers assigned to review a book. And what's more, they published the review 3 weeks before the publication date of the book. So I think they wanted to affect the discussion of the book when it first came out. So let's look at what the review had to say. And kind of interestingly, if you look at those 3 names, you look at the one at the last one, why, it's Richard Lenski. So Richard Lenski was one of the reviewers. And when I saw that, I was ecstatic. I said, yeah, give it your best shot. Tell me where I'm wrong. And so let's go through and see a couple of the points that the reviewers made. The first paragraph of the review was one in which they alerted all their readers that this was an unreliable person. They say in 1996, Michael Behe introduced the notion of irreducible complexity. And that notion excited creationists. Okay, imagine, here's a scientific magazine, and they said, this guy, creationists love him. You're not going to be well disposed towards the book. And they said that a federal judge rightly rejected this notion. And it's hard to see how a judge who was an English major in college is going to evaluate this thing if these guys can't. They haven't said anything about it. And that in Darwin Devolves, I continue chaotic efforts to overturn modern evolutionary theory. So it's nice to prime your readership by saying this is an unreliable guy and this is more chaotic discussion. Okay, well, all right, so they set the stage, but then they get into a topic of the book, which also happens to be the topic of Richard Lenski's research. And what do they have to say about that? They say, yeah, sure. There are indeed many examples of loss of function mutations that are advantageous. But Behe is selective in his examples. I select the ones that are loss of function, which are advantageous. He dedicates the better part of chapter 7 to discussing a 65,000 generation E. coli experiment. Guess who was doing that? Emphasizing the many mutations that arose that degraded function. An expected mode of adaptation to a simple laboratory environment, by the way. And number one, it was not expected. Why did they do the experiment in the first place? And lots of people before them had done similar kinds of experiments, all of them expecting to see new systems arise. And then he says, I point to those mutations and dismiss improved functions and deriding one new one as a sideshow. And full disclosure, Richard Lenski is the person who conducted those experiments. Now this sideshow accusation, he said, I called one mutation a sideshow. And the one he was talking about, here are, as an example of the many different mutations that degraded DNA and improved, he was talking about the citrate mutation. And I said in the book that, this improved things, but I said it was a sideshow because, again, as I mentioned a few slides ago, it was preceded and succeeded by loss of function mutations. So if you're losing function left and right, then the occasional helpful one isn't going to roll back the tide. Okay, he writes that about his experiment, and he writes nothing else about his experiment. That's the entirety of the review's comments on Lenski's work, even though, again, his is the best experimental evolution work that anybody has done. And yet he doesn't even mention it. And I focus on that in a large portion of the book. And again, all he does is say that he was miffed that I called one of the mutations a sideshow. They also write the following, that Behe ignores the fact that some of his prior arguments have been dismantled. He includes a lengthy appendix that argues that the blood clotting cascade is irreducibly complex, but fails to mention Kenneth Miller's simple, elegant scheme for its stepwise evolution. Okay, I've responded to all of these things, and here's my response, which is on the website of the Discovery Institute. Let me just summarize my response to it. The citation that Lenski had talking about Kenneth Miller's work was to a chapter in a philosophy in a philosophy book about design versus Darwinism. And it turns out that was a reprint of the same article that appeared 5 years earlier in a different philosophy book called Darwin or Design, which was published by Oxford University Press and had different people on different sides of the design-Darwin issue, including Kenneth Miller. Another person who had a chapter in that book was me. And in it, I addressed Kenneth Miller's objections to the blood clotting cascade and to the bacterial flagellum. So let me make explicit that Lenski and co-authors faulted me for not responding to a paper, a chapter, in which I wrote a chapter responding to it in the same book it was published, the same one. And additionally, if you go back and actually read the paper, the scheme that Kenneth Miller has for blood clotting evolution through Darwinian means is paper thin and too general to be tested. And since then, nobody has published a paper in a journal experimentally or even theoretically or computationally trying to explain how such things could arise. Here's another one. Acceptation also challenges Behe's notion of devolution by showing that loss of one function can lead to gain of another. The evolutionary ancestors of whales lost their ability to walk on land as their front limbs evolved into flippers. But flippers proved advantageous in the long run. So there. It's hard to believe that smart guys actually write things like this. They say that, okay, ancestors of whales had legs. Whales have flippers. So isn't Darwinism wonderful that it caused that? But the whole book was asked the question, is Darwin's mechanism responsible for changes in life? And they didn't even address the question. They didn't even seem to comprehend it. And they closed the review by saying, ultimately, Darwin Devolves fails to challenge modern evolutionary science because, once again, Behe does not fully engage with it. He misrepresents theory and avoids evidence that challenges him. There is a notion in psychology called projection. And projection is, of course, where you ascribe your own faults to somebody else. And I'm afraid that Richard Lenski is the one who is misrepresenting theory and avoiding evidence that challenges him. Okay, I only have a couple minutes left to go, so I'm going to skip a few slides. That's a good one. Let's go to this one. This is where he says, Lenski writes that the major problem with the book is that I'm right that mutations that break or blunt a gene can be adaptive, and he's right that when such mutations are adaptive, they are easy to come by. But he's wrong when he implies these facts present a problem for evolutionary biology because his thesis confuses frequencies over the short run with lasting impacts over the long haul of evolution. So he's saying, there can be these little minor changes, but when a big old, beneficial mutation comes along, that's what really affects life. And he gives, I keep doing that. And he gives a couple examples of this contrast between frequency and importance. He writes that common colds are very frequent, but their effects aren't so great. But if you get the Ebola virus, which is much less frequent, then that could have much more profound effects on your health. If you invested in the stock market and you bought stock in 100 companies, but 80 companies went bankrupt, but 20 companies did really well, well, you could become rich anyway. And Tiktaalik is a famous fossil that looks like it has wrist bones. And he says, well, yeah, there's lots of changes in the evolutionary record, but hey, this was a big important one because all the animals we're familiar with, they have wrists and such. And I was right. The problem, of course, is that none of these illustrations is relevant to the matter at hand. None of them has to do with evolution, let alone with molecular changes, which I was addressing. Even the cold virus versus Ebola, that doesn't say anything about evolution, even evolution of viruses. It just says one is common and one is more dangerous. And the stock market, what's that got to do with anything? And Tiktaalik, where did that come from? What are the mutations that might have allowed changes in that? Are they able to be obtained by Darwinian processes? Again, he doesn't even consider that. It's the idea that whatever happened must have happened by Darwinian evolution, and doesn't even seem to comprehend the question. Now, I forgot to mention that Lenski wrote these things not in the review, but in a subsequent posts on his blog. And one criticism of folks who were defending my book is that, well, the authors of the review only had 750 words to review the book. So sure, they're going to be, they can't get into particulars. And I don't think that's valid, but nonetheless, Lenski later on, a few weeks later, addressed the book again in 5 lengthy posts. And this is his considered view with all the space he needs, all the space he needs to address the problem after thinking about this for a long time. And this is the best that he could come up with. So I guess the long and the short of it is that it's clear, to me certainly, that nobody has an answer to the argument of Darwin devolves. Everybody admits degradation is easy and can help on occasion. And as for counter arguments, they range from poor to laughable. So I think the advance in technology that we have gotten, allowing us to sequence DNA, is really, really the death knell for Darwin's theory. And so again, I showed that slide yesterday, and the website of my new book, darwindevolves.com, contains links to all reviews of my book, plus my responses to those reviews. So anybody who wants to see the nitty-gritty can go there and track them down. Thanks very much for your attention.